The DNA isolation from human blood experiment was developed on October 29th of 2012 in the Microbiology room in the Yellow Building of Gimnasio Campestre School, Bogotá, D.C. It was performed by Pablo Bonilla, Federico Cruz, Juan Pablo Montoya, and Nicolás Revollo, with the instructions of Mauricio Jiménez and Pablo Barriga.
The objective of the experiment was to isolate the DNA of a human blood sample.
This was supposed to be done by purifying the blood with a saline solution, so the blood plasma would be removed and consequently the cells would break down to release its nucleus. Afterwards, proteins from the cell membranes are eliminated using an enzyme that degrades them. This with the purpose of making proteins heavier and precipitate them with centrifugation using a salt. Finally, the precipitate will be added with an alcohol. At this point of the process, some kind of white-colored spot is seen in the sample.
In this step, Nicolás Revollo was the one in charge of holding the tube while the blood sample was being added. After doing that, the assay tube with blood in it was put back in the tuber holder.
In this second and third step of the protocol, Pablo, our teacher, helped us adding the TE1X buffer to the sample until the established volume was reached to go on with the experiment.
These last steps were done to purify and consequently remove the blood plasma from the sample using the buffer, saline solution. This is accomplished by mixing the solution using the inversion method approximately 6 times. After we completed the last steps, we gave the sample to Mauricio Jiménez, our microbiology teacher, so he could put it in the centrifugator so the fourth step would be developed.
After the centrifugation, the plasma should be separated from the blood cells, so the plasma goes up in the sample while the cells go down.
The fifth step was to take out the supernatant. At the beginning, Nicolás Revollo was doing this step, but he got closer to the supernatant in the tube. Mauricio helped us doing it right because at that moment the precision was required and no mistakes were allowed. But before that, we had to identify the supernatant in the sample, which was supposed to be a clearer part in the bottom of the tube.
Then we did have to take it away. Then we had to add 1 milliliter of lysis buffer and then put the sample in the vortex. Mauricio Jiménez helped us add the lysis buffer to the sample and then Nicolás Revollo put it in the vortex.
This step is done to prepare the cells before the breakdown in the next step of the experiment. Then we add 67 UL of SDS and put it in the vortex. In this step, Mauricio also helped us adding the SDS and there were no physical changes in the solution. The SDS is a detergent that denatures proteins and breaks the lipids link, which leads to break into the membrane.
Then we had to add 335 UL of NaCl5M and put it in the vortex. Mauricio helped us adding the 335 UL of NaCl5M, and after that the sample developed some kind of bubbles. This will remove all the contaminants from the substance and place them in the top of the tube so they can be physically removed. Then we put the substance in the vortex.
After putting the sample in the vortex, the substance had to be centrifuged again at 4000 RPM.
Then the supernatant had to be transferred to a 50 millimeters sterile tube. Nicolás Revollo was the one that did this step, and after that we had to add 2 volumes of absolute ethanol at an environmental temperature and mix the sample in the tube using the inversion method
until we could see the DNA precipitation. Later on, the tube was centrifugated for 10 minutes at 4000 RPM. Right after that, the last step, the supernatant had to be taken away so the DNA could have been dried. The final result was a clear spot in the sample tube, which was supposed to be the DNA that we isolated throughout this entire experiment in the laboratory.